Hello, science fans. Welcome to a journey where curiosity meets cutting-edge discovery. Imagine building a car engine, not from a kit, but from raw materials. Every bolt, every gear, every tiny piece crafted by hand, with nothing but blueprints and raw elements to guide you. Now take that idea a step further. Imagine building the engine of life itself, DNA, the intricate blueprint found in every living cell, the code that makes us who we are. Today, scientists are learning to create human DNA from scratch, starting with just basic chemicals in the lab. It's like assembling the ultimate instruction manual for life, one letter at a time. This is real science, not fiction, and it's raising profound questions about what it truly means to be human, and how far we should go in rewriting our own biology. At the heart of this revolution is the Human Genome Synthesis Project, or GP-Write, a global effort to push the boundaries of what's possible. The goal? To write a complete set of human DNA instructions from the ground up, creating a synthetic genome that could unlock new frontiers in medicine and biology. This leap could let us design virus-proof cells, create new medicines, and even engineer biofuels to power our world. The possibilities are as vast as our imagination, but with such power comes new ethical challenges, questions that society has never faced before. The power to write life is unprecedented. It's a responsibility that demands careful thought and global conversation. When people hear synthetic human DNA, they worry about designer babies, artificial humans, or unintended consequences that could ripple through generations. These concerns are real and deserve open, honest discussion. We need to listen to every voice, not just the scientists. This isn't just a scientific debate, it's a conversation for everyone, from policymakers to parents, from students to teachers. We all have a stake in this future, because the choices we make today will shape the world of tomorrow. We need to carefully weigh the benefits and risks, considering both the incredible potential and the possible dangers. And together, as a global community, we must decide what rules and boundaries should guide us as we move forward. So let's dive in and explore this new frontier of science and ethics, where every discovery brings new questions. We'll separate fact from fiction and see how scientists are building DNA and why this matters for all of us. We'll face the tough questions. What ethical lines shouldn't be crossed? Who decides, and how do we protect our shared future? How do we ensure this technology is used for good, and that safety always comes first? The future isn't something that just happens, it's something we build, step by step, with knowledge and care. Understanding the science is the first step. Let's get started. Consider the following. So, what exactly is GP-Write? And why is it making headlines in the world of science? GP-Write, or the Human Genome Synthesis Project, is a groundbreaking global effort to figure out how to build large genomes, including the entire human genome, from scratch using advanced technology and international collaboration. Imagine your genome as the ultimate instruction manual for building you, written in DNA's four-letter code, A, T, C, and G. Every cell in your body reads this code to function and grow. For decades, scientists have become experts at reading that book, decoding the sequence of our DNA to understand what makes us human. But now, they want to take the next step, actually writing it, letter by letter, to see if they can recreate life's blueprint. GP-Write is the logical next phase after the Human Genome Project, which successfully mapped our DNA and gave us the first complete readout of our genetic code. But there's a big difference between reading a blueprint and actually building what it describes. By constructing a genome from the ground up, scientists can truly test their understanding of how life works at the most fundamental level. This shift from reading to writing DNA is a massive leap forward in biology, opening up possibilities we could only dream of before. It moves us from simply observing life to actually designing and engineering it, giving us new power to shape biology. The project isn't just focused on humans. Scientists are starting with simpler organisms like yeast and bacteria to develop and perfect the techniques needed for more complex genomes. These simpler life forms act as testbeds, helping researchers refine the methods and tools required for human DNA synthesis. The goal isn't to create synthetic humans, but rather to make human cells in a lab dish that contain entirely synthetic genomes, allowing for controlled experiments. These lab-grown cells can help us study diseases, test new drugs, and unlock a deeper understanding of life's rules in ways never before possible. Ultimately, it's about gaining deeper knowledge and creating revolutionary tools for medicine, biotechnology, and even agriculture. The focus is on the cell, the basic building block of all living things where the real magic of life happens. Not on creating a whole person, but on understanding and engineering life at its smallest, most essential level. To see why this matters, let's look at the incredible journey of genetics, a story that has unfolded over centuries and changed the way we see ourselves. For a long time, we knew that traits were inherited from one generation to the next, but we had no idea how this mysterious process actually worked. The discovery of DNA's double helix in the 1950s changed everything, revealing the elegant structure that underlies all living things. Suddenly we had the alphabet of life in our hands, a code that could explain the diversity of every organism on Earth. The Human Genome Project then set out to map all 3 billion letters of our DNA, a monumental task that took over a decade and involved scientists from around the world. This gave us a detailed map of our genetic landscape, but not a full understanding of what every part of it means or how it all works together. We know what some genes do, but much of our DNA remains a mystery, vast stretches whose functions we're only beginning to guess at. Now, GP Wright changes the game. Instead of just reading the map, we're trying to build the country from scratch, designing and assembling DNA with intention. We're moving from being genetic readers to becoming genetic writers, taking a more active role in shaping life itself. This is like going from listening to music to composing your own symphony, creating new possibilities that never existed before. Writing a genome forces us to understand its structure and meaning deeply, pushing the boundaries of our knowledge and creativity. If we put a gene in the wrong place, what happens? Could it cause a cell to malfunction, or might it reveal something new about how life works? If we change a sequence, how does the cell respond? Each experiment is a step into the unknown, with the potential for both breakthroughs and surprises. These are the questions GP Wright aims to answer, as scientists collaborate to unlock the secrets of our genetic code. It's the ultimate test of our knowledge, a challenge that pushes us to the very edge of what we know about biology. By building DNA from the ground up, we learn what really makes life tick, and what's possible when we take control of the code. This is biology's next great chapter, a leap as significant as the discovery of DNA itself. The shift is profound, from observation to creation, from decoding nature to rewriting it. The possibilities and the questions are enormous, opening doors to new cures, new organisms, and new ethical dilemmas. Let's see how it's done and explore the future we're building, one letter at a time. So how do you actually build a genome? It starts with DNA's 4 bases: adenine, thymine, cytosine, and guanine. Scientists use machines called DNA synthesizers to string together short DNA sequences. These machines are like high-tech printers, but they can only make short strands. To build something as big as a human genome, scientists stitch these pieces together. They use yeast cells to help assemble longer DNA fragments, like putting together a giant puzzle. Step by step, small fragments become larger chunks, then entire chromosomes. The human genome has 23 pairs of chromosomes, each must be built one by one. Once a synthetic chromosome is ready, it's placed into a human cell with its original DNA removed. If the sequence is right, the cell should function and grow using the synthetic blueprint. This process is slow, precise, and full of trial and error. The final test: can the written code bring a cell to life? It's a technical marvel, and a huge challenge, but it's happening now. Why take on such a massive project? The rewards could transform our world. In medicine, we could design human cells resistant to viruses, potentially making us immune to diseases like HIV or the flu. We could build lab-grown human cells with specific mutations to study diseases and test drugs more accurately than ever before. This could speed up cures and reveal how diseases work at the cellular level. Beyond health, we could engineer microbes to produce clean fuels or medicines, or even clean up pollution. Writing DNA lets us program biology to solve big challenges, and there's the quest for knowledge. By building genomes, we can finally learn what mysterious parts of our DNA do. It's about understanding life at its most fundamental level. The discoveries could reshape science for generations. The potential is vast, the risks are real, but the possibilities are too important to ignore. Let's look closer at the medical potential. Imagine growing transplant organs from a patient's own cells. No more waiting lists or rejection. Synthetic genomics could let us remove harmful viruses from animal organs, making them safe for human transplants. We could also fix genetic diseases at their source by designing therapeutic cells with corrected genes. These cells could provide permanent cures, not just temporary treatments. This is the dream of regenerative medicine. Synthetic genomes also help us study aging, why our bodies break down, and how to extend healthy years. We could create cells resistant to aging or disease. This research could help us fight pandemics too. By quickly synthesizing viral genomes, we could speed up vaccine and drug development. The impact on health and longevity could be enormous. The science is still young, but the promise is real. The future of medicine may be written in synthetic DNA. With great power comes great responsibility and concern. One big worry is safety. What if a synthetic organism escapes the lab? It could harm people or the environment if not properly contained. There's also the risk of misuse. Someone could engineer a dangerous virus as a weapon. The same tools that cure disease could be used for harm. Then there are ethical fears, like the idea of designer babies or a genetic class divide. Some worry this technology could deepen social inequalities. Others feel it crosses a moral line, playing God with life itself. These are not just scientific questions, but societal ones. We must address them together. The phrase "playing God" captures a deep unease. It's not just about accidents or misuse, it's about whether we should rewrite life at all. Some fear we can't predict the consequences of changing a genome. Others value the natural world and worry about blurring the line between natural and artificial. There's also the risk of genetic inequality. Will only the wealthy benefit? The scientific community is taking these issues seriously. Ethicists, policymakers, and the public are part of the conversation from the start. The goal is to set rules and guidelines before the technology races ahead. Open dialogue is crucial. We must balance innovation with responsibility. The debate is ongoing and essential. So how do we move forward safely? The answer: strong guardrails. Lab safety is key. Synthetic organisms are engineered to survive only in the lab. Oversight and regulation matter. Companies screen DNA orders for dangerous sequences. International agreements are needed to keep standards high everywhere. But rules aren't enough. We need a culture of responsibility among scientists. Ethics training and public engagement are essential. The GP Wright Project is leading by example, prioritizing ethical and social issues. Ultimately, an informed public is the best safeguard. The future of this technology should be shaped by all of us, not just scientists or politicians. We must learn, discuss, and decide together. By staying engaged, we can ensure this power benefits humanity. The journey is just beginning. Let's take it together.